Thank you very much for the nice introduction. And today I will talk about how to come from a metabolomics and analytics zero to metabolomics hero within only 2 days, because this is possible when having nice instrumentations from HLand in combination with a standardized assay as we have it as biocrates. So let's have a look at this. And I will talk about two parts. So first we will have a brief look at targeted metabolomics and its applications, and then we will focus to one of our kits, the Quant 500 XL kit. So let's get started with targeted metabolomics. What is biocrates? We are a company for targeted metabolomics and are located in Innsbruck in Austria. There's our headquarters and a subdivision in United States. Our kits we have, they are mainly for triple quad instruments, like the HLand 6495 series. All results which are coming out of kits are quantitative and standardized. And there's worldwide a global community with over 100 kit users who are happy to use this technology. What is the advantage of standardized assays as we provided as biocrates? So one of the big advantages is that the results are in the end reliable, they are robust, transferable, and reproducible. So what does this mean? When doing analysis or profiling and you develop assays on your own, which is definitely a nice approach, but if you do this, you need several persons for this. And it takes some time. When using a standardized assay, this can be done with only one person and a robust instrument, so it is cost and time saving. And the advantage is you can run samples on multiple instruments, so let's say on 2 or 3 of these HLand instruments in parallel, so you can scale up the analysis and the results are still robust and comparable. There are different applications for metabolomics. Let's just have some examples here. So in pharma, you can support or to enable personalized therapies by doing metabolomics. In epidemiology and biobanks, for example, for population-based studies. So if you have got cohorts, it can be easily used to identify, for example, risk factors or to improve individual health. So here you can scale up the analysis easily by using a standardized assay. And it's also nice for nutrition and well-being. For example, it can be analyzed how eating habits and lifestyle have an influence on well-being and the health and the condition of an individual person. A wide range of matrices from multiple species are well suited to be analyzed with our kits. We have got standard operating procedures, or let's call them protocols, available for multiple matrices. If you're going to analyze one of them, just reach out to the support team and then we will help you. Either we have got a standardized procedure for it, then it's quite easy. You can directly use it and you're ready for analyzing those samples and to do metabolomics with it. Or if you have got a matrix where no standard operating procedure is available, then let's have a look at this example here from a laboratory using HLand machines in Poland. They wanted to analyze human aqueous humor, and first they made a test. They tested the matrix, they optimized the protocol for the sample preparation, and also they tested different volumes which are analyzed with the kit. After this easy test, they identified, for example, the best sample volume, and then they ran their study and published their results and could prove their key hypothesis. So it's quite easy to do it. It's fast. because the assay, we will see this soon, the assay is standardized, and then there's no time which has to be invested in developing the assay. So let's have a look at the Quant 500 XL kit, one of our standardized assays. It is available for the Agilent 6495 instrument, so the D instrument. The sample volume is 20 microliter for serum plasma and also other biological matrices, so a small sample volume is sufficient for analyzing up to 700 metabolites. For example, for another matrix, feces, 200 milligram are required. The assay is validated for human plasma. Other matrices are tested, like feces, red plasma, and so on. This we will see soon. The kit is coming in 96 well plate format, and with each of these kits on each plate, approximately 79 samples can be run. Why approximately? It depends a bit on the study design, how many replicates of standard samples are analyzed together with the study samples. Per week, up to 2 to 3 kits can be run on one instrument. And the run time is 54 hours. Please don't be scared. It's not hands-on time. Most of this time is instrumental running time. More metabolites are included in the assay, then they can be detected. For example, for human plasma, approximately 720 metabolites are above the limit of detection. Multiple small molecules from different classes and a wide range of lipids are detected by the Quant 500 XL kit. And if you are interested in the list of metabolites, please feel free to reach out to us, then we will send you a document where you can find all the metabolites which are covered by the kit. And then there's a very nice feature with all of our kits, in addition to the pure concentrations of each metabolite which is covered, so-called metabolism indicators are calculated. These are indication-driven sums and ratios which are proved by a publication, for example, for aging or for diseases. And they are calculated in addition to the concentrations and can be added to the results at the end of the quantification. So let's have a look at, as an example, to one of these metabolism indicators. So there is no need to develop all of them new. There are old ones, for example, the Fisher ratio, which was published in the 70s, so a long time ago. It is an indication for liver failure, and for sure can be, if it matches the study design, can be included into the evaluations. And the choice is yours to include those metabolism indicators into your research, which are provided with the kit. The assay itself, the robustness for sure, is always tested by biocrates, and here we see an example of interlaboratory robustness. The same set of samples was run in different laboratories, and then the results were compared. So the concentrations from all metabolites measured in all laboratories were compared, and for plasma samples, on average, the variations are below 9%, for feces slightly higher. So what does this demonstrate? Results are highly transferable and comparable. You can run samples on different machines, also in different laboratories, especially when you've got a cohort. You can split this maybe to 2 or 3 laboratories. They can run the samples and the data can be combined in the end. How does the workflow look like of our assays? The kits are coming together with our so-called WebIDQ software. It's a workflow manager, and it supports you during the entire process of the kit use. What are one of the key features? The the samples are registered in the software, data is processed, evaluated, then there are the metabolism indicators, and normalized results are exported in the end. So let's have a closer look at this. At the beginning, you've got your project, you've registered the samples, and then there is a layout of the well plate which you design in the software. There's a sequence which is exported for the Mars Hunter software. So there is no need to write the sequence for all these injections manually. You export it and directly import this into the Mars Hunter software. And the performance of the agent machine is tested. We will see this soon. And if the performance is fine, then the kit preparation starts. So in any case, if there should be any challenge with the system, which can always happen, you will not lose your samples. You can do troubleshooting, for example, cleaning of the ion source of the system. Once everything is fine, then you start with the sample preparation. Then you run it with your HLN system, and the result, so the MS data files, so the D files, they are then transferred into the WebIDQ software, where data processing, which means integration, quantification, calibration, and evaluation is performed. In the end, after data preprocessing and normalization, you've got a standardized export with your results. What is included in the kits? In the kits, for sure, not everything is included. For example, the mass spectrometer system is a requirement which should be there, or pipettes in the laboratory, but all key things which are required for the assay to keep it standardized, they are provided with the kit by biocrates. There's an analytical column which is provided, and also the gradient is given. So you just use this gradient and everything is fine. Only retention times have to be adjusted when using the kit for the first time. Kit plate is included, samples for the system suitability test to check the performance of the LCMS system, calibration standards and quality controls. The instrumental methods, they're ready to use, no tuning or optimization is required. You copy them to Mars Hunter and you can start. Comprehensive documentation and support. So whenever you will have got a challenge, or an issue, or a question, you can always reach out to the support team and we will help you by email or video sessions. And for sure, biocrates WebIDQ software, which works then in parallel with Mars Hunter. The sample volume is small with 20 microliter, and the workflow is easy with WebIDQ automated, and the setup in the laboratory, so when using the kit for the first time, takes approximately 2 days, and then you're ready to run the full kit. The sample preparation is performed on a 96 well plate, which is coming with the kit. When setting up the methods for the first time, the instrumental methods, you copy them to Mars Hunter. There's no tuning optimization required. This procedure is assisted by the customer support, and there is for sure also a training included when starting for the first time. For testing the methods for the first time and also for testing the system, so-called test samples are provided. They are ready to use vials, and metabolites of the assay are included. You run them, you have the chromatograms, and you can check if the performance is fine or not. There are 3 of them because there are 3 parts, analytical parts, and for each part there's 1 test sample. Here I will not go into much detail of this. The plate layout, a default plate layout looks like this. We have got a blank, we have got 0 samples where internal standards are included, but no matrix. They are to determine the limit of detection. So the limit of detection is calculated for each kit plate specifically, calibration standards and quality controls, which are provided by biocrates. If study, specific quality control samples are available, for sure the biocrates quality controls can be replaced by study quality control samples like pooled plasma, for example. They are used, these quality controls, to check the performance of the kit plate and also to do interplate normalization. And with the standard layout, 79 samples can be analyzed. The layout can be really adjusted to your needs. The layout is designed in the WebIDQ software. In the end, an export is created. You get a printout for a laboratory for preparing the kit plate and also for running the sequence with Mars Hunter. The preparation of the assay is quite easy. You pipette 10 microliter of the sample on each kit plate. It's dried down, derivatized, dried again. Metabolites are extracted, eluted, and then there are three 96 well plates which are provided with each kit, and 3 different dilutions are done for different analytical injections. There are 6 injections with the Quant 500 XL kit, 2 in the so-called LC part where chromatography is used, and 4 in the so-called flow injection analysis FRA part where no column is used. In the end, all MS data, so all DIF files are imported into the WebIDQ software and processed. And then, after processing the data in the WebIDQ software, which is quite easy and streamlined, all results are quantitative, so they are given in micromolar. And then there's a quality control normalization, so a so-called recalibration for all metabolites, so that you have got highly quantitative results for all metabolites which are above the detection limit. The performance of the assay is automated checked in the WebIDQ software, and a color code guides you if there is a need for double-checking the performance of an analyte in a specific well, for example here at the blank well, or if everything is fine, then it's green. And whenever there are questions, the support team is always looking forward to assist you. So in summary, what are the advantages of having a standardized assay and running them with an HLint 6495? You can get started easily, rapidly, so without delays. No method development is required. The kit setup is done in 2 days for different matrices, sample matrices, SOPs, so protocols are available. There is also a reference database available if this is required. You can start easily and rapidly. You can scale up the analysis to a large number of samples. The quality of the kit was tested. All results are in micromole per liter, and the workflow is highly standardized. And the biocrates support team and all other members of biocrates are always on your side and are looking forward to helping you and to assisting you with your project and your analysis. So thank you for your time and interest.